Microbodies. So bodies we know, or it is a small uh, circular or spherical organelles. It is also known as the microsome. Okay, so these are very small, almost spherical organelles, and their diameter usually ranges from 1 to 2 micrometer, which is bounded by the single membrane. Basically, there are two type of the microbodies, and these both are present in the animal and the plant cell. And the main type of the microbodies are the peroxisome and glyoxysomes. Okay, glyoxysomes. So these are the two types of the microbodies. So peroxisomes, they usually occur in the green cells, and they contain the oxidase enzyme, especially the glycolate oxidase, which catalyzes a reaction which is important in the photorespiration. So let's see what are the different. Yeah, this is the diagram of the microbodies. As we can see that it is, surrounded by the single membrane. The internal compartment is known as a lumen, which consists of the different digestive enzymes. Some proteins is there. Okay, some enzyme substrate complexes are also present there. Okay, so we can see through the diagram, and this is the diagram of the peroxisome. It is just a same diagram of the peroxisome as well as the glyoxysome. They have the lipid membrane, which is a single membrane, and a matrix is present, and the internal core is present, which consists of the many digestive enzymes, protein, and several enzyme substrate complex. So these are also refers to as the cytosomes, and the microbody is a typically spherical vesicles with a diameter of between 0.2 to 1.5 micrometer. The microbodies, they can be seen in a cell cytoplasm, and however, they can only be viewed with the electron microscopy. Okay, so they can be not visible by the single, means a simpler microscope, but they can be seen in the electron microscope. They are surrounded by a single phospholipid bilayer membrane and have an intercellular protein and the enzyme matrix. This is structure we are talking about. Okay, so they have the protein, we can say, or and the enzyme matrix, but it does not appear that they have any genetic material that would allow them to self replicate. So let's see what are the functions of this microbody. So microbody are the cell organelle which are present in both the plant as well as the animal cell. Okay, so these cells are present in both the animal as well as the plant, and typically they are found in the cytoplasm only, and they are the vesicular and mostly spherical in the shape. Okay, and the various type of the microbodies usually exist, and they perform the several functions within the cell. The first one is the biochemical reaction. Biochemical reaction means microbodies, they play a very crucial role in the various biochemical reaction within the cell. Enzymatic activity, the enzymes which are present in the microbody, they facilitate the essential reactions such as the breakdown of the fat, breakdown of the amino acid, as well as the alcohol. So that function is performed by the microbodies. The photorespiration in plant microbodies, particularly peroxisome, they are involved in the photorespirations in the plant and detoxification. Obviously, microbodies are also responsible for the detoxifications of the peroxide and sometimes most of the synthetic chemicals. So, the functions of the microbodies is in the detoxification also. So, let's discuss the two type of the microbodies. The first one is the peroxisome, and second one is the glyoxysome. So peroxisome, it is identified by the Christian D. Dewey in the 1965, and it is discovered using the fractionation method. And it is present in both the plant cell as well as the animal cell, but they are particularly prevalent in the liver as well as in the kidney cell. Okay. They are also found in the close proximity to the other organelles like the endoplasmic reticulum, mitochondria, okay, endoplasmic reticulum, mitochondria, and chloroplast. So they are found in the close proximity of these organelles. Okay, the origin, the endoplasmic reticulum gave rise to 70 to 100 peroxisomes in the photosynthetic cell. They are vary in the size, their shape, and in the diameter, and they have the membrane bound organelle using means we can say with an outer membrane only, and which are with a covering of the granular and a fibrillar containing internal matrix. Oxidative enzymes are present in them, in the peroxisomes. Oxidative enzymes such as the urate oxidase, hydroxyoxidase, and the catalases. So these all enzymes are present in the peroxisomes. And the catalases, what actually catalase do? Catalase rapidly metabolize the hydrogen peroxide H2O2, which is produced during the oxidative processes. So identification, the peroxisomes usually identified using a catalase biochemical reaction, and the larger peroxisomes, they can also be observed in the hepatocyte. Okay, in the laboratory preparation, so a structure can be visualized in the stain preparation, which are appearing as a minute organelle within the diameter, and the urate oxidase, it is found in the nucleate and the crystal structure of the core. So the functions of the peroxisomes are that they are the membrane bound organelle, which we know that the single phospholipid bilayer is present, and they are present only in the